Chujan Chatterjee, if you can hear me, could you please turn off your video so that we can preserve the bandwidth? Thank you.
Hello.
Yes, sir.
Yeah, I'm audible, right? I'm audible, all right?
Yes, yes, you're audible.
Oh,
We'll
cool.
start. Yeah, we'll start in about 10 minutes.
That's okay.
Yeah, yeah.
Yeah.
Sir, if you could, you were presenting your title slide. If you could.
Yeah, I'm
Do
going to do that. I just wanted to make it ensure that I'm audible. So
Yes, sir.
yeah.
Yes, sir. You are. Thank you. Thank you.
Okay. Yeah. Cool. I'm going to do that right
Okay. Thank you so much.
away.
Welcome to our Progress and Prospects in Biology webinar. This is our webinar number 17. We'll start for today, but before we begin, just a few housekeeping rules like every week. So just keep your microphones on mute and turn your videos off so that we can preserve the bandwidth. And also when the speaker is presenting, pin the speaker slide to your screen. Having said that, please do not share your screen when during the presentation or at any time. So these are just a few rules. So if everybody could maintain that. With that, I'd like to welcome today's speaker, Dr. Arijit Basu, who's a senior scientist at Takeda Pharmaceuticals at Cambridge, Massachusetts, in the USA. So I would now turn on to our convener, Professor N.R.A. Banerjee, to welcome the speaker. Ma'am, I'm not sure, is ma'am here? Ma'am, if you could welcome the speaker before I go on to introduce him.
Yeah, thank you, Sanjana. Just before I was about to, respond to your request, my connection got lost. So we panicked for a while, but we are back. So a warm welcome to Arijit, and I hope Mohuadi is here. So we are set to begin, and a warm welcome to all the participants. A few words about the webinar series, Progress and Prospects in Biology. And I think one of the main purposes of the webinar series is to sensitize students, researchers, professionals, and others who are logging into our talks regularly, is about the varied spectrum of topics that people are working on and the possibilities of extending beyond the discipline that one has trained in. And I think what better than Arijit's talk today to introduce one to that. And Arijit, I listened to your last talk that Mohuadi had shared with me, and that is when I got inspired that I must have you speak on our platform, because it is in your talk that you speak very passionately about how different disciplines converge and how you have been motivated to train in different disciplines that you had not during your studenthood. So I would request Sanjana to introduce our speaker today, and I think we will reserve our whatever is going through our minds, our comments and questions till it is over. So I think we are ready to listen to you, and Sanjana, you may want to introduce our today's speaker.
Thank you.
Thank you, ma'am. Thank you so much. And like I agree too, like it's all about interdisciplinary and multidisciplinary research. So thank you, sir, for being here. So today we have Dr. Arijit Basu. Dr. Arijit Basu is a senior scientist at Takeda Pharmaceuticals working on computational modeling and evaluation of drug safety. After completing his bachelor's in pharmacy from Baharampur University and master's in pharmacy from IIT Varanasi, Dr. Basu earned his PhD in medicinal chemistry from Birla Institute of Technology, Ranchi. He was also an assistant professor at BIT Mesra, following which he held a research scientist position at the Hebrew University of Jerusalem. He then moved to MIT and was a postdoctoral research associate at the Koch Institute for Integrative Cancer Research. Dr. Basu has published 44 peer-reviewed journal articles and 11 book chapters. He's the associate editor for the Wiley Journal, Polymer for Advanced Technologies, and he is a recent recipient of the Young Investigator Award by Polymers for Advanced Technology Society in 2019. We are so happy to have you here, Dr. Basu. Thank you for being here. just before I begin, Pravat Kumar Swain and Sanjay Tiwari, if you could please turn off your video, it'll help us preserve the bandwidth. And with that, over to you, Dr. Basu. Thank you for being here.
Well, thank you, Sanjana. Thank you, Professor Banerjee, for this kind and lovely introduction. I hope I can actually keep up with that expectation. And yeah, I completely agree with Professor Banerjee and Sanjana that we should not be limited to our research field, and we should learn and do whatever is needed for a project to succeed. Being said that, there is also another caveat that we can actually learn things quickly, but in order to be expert in some field, it takes time. So it's also important to collaborate with the peers, like collaborate with the scientists who are actually working on this on the particular field for maybe more than a decade or maybe 2 decades. So that is also important to keep yourself open and want to learn and also to collaborate. So being said that, I would like to start my talk. And my talk today is focused mainly on students, mainly on undergraduate students who want to take drug discovery or drug research in general as their future career. So being said that, I will keep my talk as simple as possible. And if you have any questions, any kind of doubt or any kind of feedback, please let me know. Uh, so, so I am a designer. So I mean to say, I design molecules. So I am the person who is actually responsible to start a molecule in the first place. So if something goes wrong, I am the person who is responsible, and people will come and tell, "Hey, you designed this molecule, did not work." So that is what I actually do, and I am kind of I listen to many scientists from all corners. So drug designing, as we know, is an extremely complicated process, and I will try throughout my talk to convey that even more. So with that, I will start with the first slide, kind of, or sentence. What's a drug? So it's uh, it's we all know kind of what's a drug. I mean, I'm sure there is no one in the audience that has never taken a drug. So we know drugs are actually, by definition, drugs are actually a chemical substance that produces a biological effect. But in this talk or in this particular topic, we are actually talking about a biological effect that is actually beneficial for for either prevention of a disease or a cure of a disease or mitigation of a disease or maybe diagnosis of a disease. So we are talking about therapeutics here, not drug as such. But what are they? So drugs are actually a chemical substance. So it's a chemical moiety. It can be an organic chemical. It can be an inorganic chemical. Though 95% of our drugs are organic chemicals, very few are inorganic. And it has to have a biological effect. Then it is called a drug. This effect may be beneficial. This effect may be not beneficial. That's a separate question. But our talk focuses about those chemical substances that have a beneficial effect on our biology. And that is what we call a therapeutics. Now, we know a few drugs, of course. Drugs are chemicals, so the chemicals have chemical structures. And so this is aspirin. It's a very small and simple chemical, which is acetyl salicylic acid, and it looks very simple. So we all heard about it. We have possibly most of us have taken aspirin, or many of us may have been taking a slow release aspirin in the form of ecosprin or something like that. And drug molecule can be very complicated in structure, like Taxol. Taxol is an anti-cancer drug that is derived from a natural source, and it can be an enormously complicated molecule. And and it's very difficult to understand how nature synthesizes these very complicated chemicals. And some drugs, of course, they make us feel, and if you take it, you're in a different world, and so it's addictive. And of course, we are not talking about this, of course, but definition of a drug, this also fits. Even nicotine, the cigarette smoke, nicotine is also kind of a drug because it causes a biological effect, same as cocaine. And who doesn't know this drug? I mean, this,
So,
yes.
Sorry to interrupt you. I was just wondering, people are saying in the chat that your audio is not very clear. So I don't know, is it if your microphone is like rushing against something, if you could check. Sorry about that.
Can you hear me now?
Yes, yes, yes.
I think I have to be a little bit close to the laptop, so that might be a reason. Yeah.
Okay, okay. Yeah, so people couldn't hear you clearly, so I had to interrupt. Sorry about that.
That's okay. That is perfectly fine. You do whenever there is a problem with the audio. So
Okay.
yeah, so
Good
I
luck.
think I'm stuck in the right slide. So I mean, everybody knows this guy. This is alcohol, and this is absolutely good for any mood, as per the people who use it. I mean, but in general, it's a CNS depressant. So so these are different molecules and their molecular structures that actually can form a drug. But things have changed. We are talking here about therapeutics, and we are not restricted today in small organic molecules. Today, therapeutics have reached to enormous levels, and today therapeutics can be anything. It can be a cell. Hello.
Yes, sir. Everyone, please mute your microphones. Everyone but the speaker, please. Yes, sir. Someone probably unmuted their microphone.
Okay, that's okay. It can happen.
Okay.
So drugs can be, therapeutics can be anything today. It can be an RNA molecule, which is a ribonucleic acid, and it can be a DNA. It can be antibodies. Antibodies are a hit now. It's like so many antibody drugs that are coming up. Recently, with the approval of CAR T cells for blood cancer, it is also like the cells as is, the whole cell can also be a therapeutics. And there are a lot of stem cells research and other things that are going on. So cell therapy is something really, really exciting. And so many companies at Cambridge are actually completely focused on cell therapy. Viruses as these also, like AAV virus and other viruses, the complete whole virus is also considered a drug today. And they are mostly used as gene therapy vehicles, but of course, so it has changed. It has changed enormously from the old days where we have natural molecule extracted, and then we have a structure, and then it is repurposed for a drug, to what we see today. So drug paradigm has entirely shifted, and today drugs are discovered in a different way, in a different manner, and it's according to my understanding, it is much smarter and much better today. So let's talk about the thing that you will find it in every drug discovery journal paper or every drug discovery talk. So everybody will have a different figure. So I also have this figure taken from somewhere. So drug discovery takes time. So the main aim of this slide is to show you. I will actually talk a little bit about this in the subsequent slides. But drug discovery takes a lot of time, and it's complicated, and it's very, very uncertain. So this is how a funnel of drug discovery looks like. Ultimately, you'll have one drug in the market that will have a associate cost of around 850 to 900 million dollars, and it will take somewhere around 10 to 12 years at best condition, in best case scenario, and which will have involvement of more than 100 scientists throughout. So getting that molecule in drug as a drug is really, really something of one of the most enormous challenges we face in the modern world. So we all appreciate that drug discovery is actually uncertain. Over 96% is the failure rate from a program. That means if you have a running program in a company, so 100 of those programs, so if you have 96 of them will fail. And the main problem of drug discovery is they can fail really, really late. So you have spent $700 million and it failed. Or sometimes a drug is out in the market and then uh, drug monitoring says that the drug has some problem with some patients and then it can also fail. So you bring a drug worth $1 billion and then it has to be withdrawn from the market. So it's an enormous gamble, and you have to really wait for the outcome of the gamble. So that is something very, very uncertain. It is super difficult because you have the best minds in the world working on drug discovery. More than 100 PhD scientists work typically in one drug discovery program, starting from design to development to discovery to development to clinical trial, and of course it is extremely difficult, and a lot of people have to come together and to get a drug out. It's a lengthy, of course. It is 12 years. Is a rational drug designing program. So if it is a serendipity, nobody knows how many years it's going to take. Of course, complicated. I'm going to talk about the complication throughout the talk, so I'm not going to talk much here. It is expensive, so because it's like close to $1 billion, $850 million is an average. So, say for example, Pfizer, which is I think the largest pharma, is its net worth is $57 billion. So Pfizer, the biggest pharma in the world, can afford 50 drug programs at a time, and before it gets bankrupt. So you can understand how expensive it is, and so yeah. So with that being said, I will start with the main core of the talk, that is how are the drugs discovered. So this is, I will try to keep this complicated science as simple as possible. Especially, my aim is to encourage the students. Um, it is difficult, but it is exciting. And since there are a lot of failures, you always have room for improvement. So with that, I will start with the core of the talk. That is, how are the drugs discovered? And before I start delving deeper, I'd like to highlight the most important biochemical reaction. I think that this is the only biochemical reaction that is important, which is that is called a molecular recognition. It's a fancy name. We scientists always like to use fancy names so that others don't understand and keep our job market hot. But what is molecular recognition? Molecular recognition is simply two molecules coming together and staying together for a few milliseconds under physiological condition. So if you have a cell, which is a physiological condition, and two proteins that can come together, associate with each other, and stay together for a few milliseconds, then some biological process or some biochemical process may be initiated. So that is what a molecular recognition is. And this reaction principle we always use to discover, design a molecule. So this is something we should make it clear at the beginning of the core of the talk. So this is kind of a typical simulation of a molecular recognition process that is happening. This small guy is maybe a small molecule drug, and this moving blob over here is a protein. So this is how a protein is actually engulfing a small molecule. And this is actually the small molecule is coming close to the protein, associate for some milliseconds, and then dissociate off. So this is what happens in a biochemical process. And this uh, interaction, we call it as molecular recognition, when one molecule recognizes the other. And so the molecule to be recognized has to be complementary. And so this is we can simulate today in computers, and using high-end computational techniques, we can actually design this kind of molecules that will be recognized by the other. And so that forms the core of drug design. Um, so before I come to the drug designing part, I would like to tell like what is the cause of the diseases. So we have like billions of biochemical reactions taking place while I'm talking to you. Possibly a trillion. I don't know the number, but I'm just putting up a number. So anything can go wrong. And if one biochemical reaction goes wrong, it can lead to some disease. Our body do have a repair mechanism, of course, and that repair mechanism can fail sometimes. Sometimes a biochemical reaction will yield in high expression of a protein that can cause a problem. There can be some mutations along the line, and that can also be cause of problem. But overall, any biochemical reaction pathway that goes wrong can lead to a disease. And I will talk about one biochemical pathway. I will not talk about that, of course, but I'll just show a biochemical pathway. So biochemical pathways are represented using this very nice figures, and and it has involvement of different proteins. This is one protein. This is one protein. So this and this protein are associated by a molecular recognition that I just showed you before. This protein and this protein will be associated with molecular recognition, and finally, it can if this pathway is actually is initiated, then possibly it is associated with a disease, and that may be cancer. So this is a cartoon that I'm going to represent throughout my talk, and showing it as a receptor that is a protein that is responsible, and that can actually cause some activation of a biochemical pathway that can lead to a disease. So we can, in general, we can forget everything, and we can say, okay, this guy, if it is activated, can cause some disease condition, maybe cancer in this case. And so our common sense says, why don't we stop this guy and then cancer will also be stopped? Yes, that is what we exactly do. So we use an agent that will stop this receptor we say, which is basically a protein, and that is stopped by a drug or a therapeutic agent, a therapeutic intervention. It can cause one, or it can stop many, and then finally it will actually mitigate the condition that is not desired. And so that is what actually happens. This is the simplest way that a disease is caused, and the simplest way to show a drug how it actually causes an intervention to a biochemical pathway. But it has a lot of caveat, and I'll talk about that. So the main one of the important thing is the specificity of your drug molecule. It should bind to one particular receptor. It cannot go and bind to anyone, and that is called the problem of specificity. So this is a lock and a key. So you will find this throughout in any biochemical text that a protein is a lock and a ligand is a key. This is a very, very simple explanation of a protein and a ligand, or a protein and a drug interaction. This is, I think, very, very simple, too simple. And sometimes we can think that this is very simple task. But we have to understand, if we just look at the perspective of a lock and a key, so this lock is inside a physiological buffer, and this lock is actually a flexible, a dynamic lock. And this key is also a flexible and a dynamic key. So you are kind of trying to find a key that for a very dynamic, for a very moving keyhole that actually changes over time. And so your key has to also change over time. And so this is kind of, I say, finding a flexible key for a flexible lock. This is in physiological condition. So this is the actual problem, and this is the actual way to define lock and a key model. But anyway, so this key should fit this lock, and it could not fit the other locks. Now, for a lock and a key model, this is quite easy to achieve. But for a biochemical perspective or a biologic perspective, this is quite difficult to achieve because a drug molecule often binds to other keys, other locks, and that can also cause some problem. So this is a problem of specificity we should always keep in mind. Um, I come to the drug discovery workflow in a practical workflow that happens in a pharma world, not in an academia. Of course, academia it's completely different, and often the early stage here it starts with academia, to be honest. In any drug discovery program, you have like three stages: the early stage, the discovery stage, and the developmental stage. I don't know much about the developmental stage because I have never been involved in developmental stage. So I will today talk about most of the discovery stage and a little bit about the early stage. So early stage is when the ideation begins, when uh, your project team will decide like what is the target that we are going to target. In fact, to discover a drug, and so that is the most important stage. That is the birth of the project, and so I'll come to that. So I'll come to the first, the early stage. I'm not going to talk about the developmental stage much today. So in the early stage, it's extremely important. People previously used to not pay much attention to the early stage problems. But today, early stage, that is target identification, target validation, and target safety. These three things are extremely important in today's drug discovery because you have learned from the past that if your target is not a safe target, that is, if the target itself causes some problem, then it leads to attrition of the drug that is already there in the market. There are so many glitazones and others that are used rampantly before, but it's no longer used so much because they causes a lot of issues. One of the major issues are liver failure and cardiac failure. Um, so, so glitazones are not a favored molecules today, and PPAR, that is the target for them, is not a favored target today because it has some problem in itself. So if you activate it, so there is a biochemical pathway that can cause downstream some problems. So we cannot target those kind of receptors. So you have to find a very good target that is quite clean and that is responsible for the disease itself. And it should not cause toxicity just by changing its biochemical function. So these are the three important things that we have to uh, keep into account. I will use an example throughout my talk, and I will take one of the first kind of rationally designed molecules, which is imatinib, and this is against leukemia, and I will start with like how the target is identified and validated. Though the target safety did not exist that time, so it has some problems with the target safety. So, so I told you about the receptors like this, but in general, in practice, when you look at the receptor or the target, it looks like this kind of a clay cast that you can see. It's like the surface of the protein. You can also represent it like this. These are the secondary structures of a protein, if you want to know. And this sometimes I was working on a laptop on my computer, and one of the engineers who were working on the lab came and told me, "Oh, you make noodles on the computers." So these noodle-like things are actually proteins. So that was a funny comment I like to share. But I'll talk about imatinib, which started discovery phase started in 1990, and it's approved in 2002. And so I'll start with an example like this, and then I'll try and explain it every step. This is the structure of imatinib, which is kind of an elongated structure. And so I will come to the discussion like how the structures have the project has come up with this structure. So we have, as I told you, that this is a moving key and a moving lock. So you can see this is actually what happens in, if you see this, this jiggling, wiggling motion of a protein is actually what happens in the physiology, and this is in I think 100 nanosecond or 1000 nanosecond range. That this is the movement that happens, and this small guy over here is the drug molecule, or is the small molecule that actually is recognized by this protein. So it jiggles around a little bit, and then it's finally recognized here, and then it goes away again. So this is what happens in a normal biochemistry, in a normal molecular recognition reaction. So this kind of reaction we represent it like this, and this we represent as a log, and this tiny guy over here we represent as T. So this is the idea. And so as I told you that a receptor that I presented as cartoon is actually looking like this. A drug which is presented as a ball, it looks like this. And then when they are coming together, they look like this. So if you see it here, this is a kind of a tunnel. It can go one, it can actually, every protein has a different kind of active site. But for this is a tunnel. So it goes through, and this tunnel is kind of a hallmark for every protein, and you have to build a molecule that is complementary to this tunnel. So in a very simple sense. So this slide I will come again and again. This is the timeline for discovery of Glivec. It started in 1990. It ended in 2002. So from 1990, it started much before. We have a protein structure. So this is very important. We should have a structure of this protein solved. These are the guys who are called structural biologists, and they use sophisticated methods to solve the structure of the protein to enable designer like us to design a molecule which is complementary to this protein. Then it's all the synthesis and everything has finished in 2 years, which is an exceptional speed. And then its biology took like around four more years. That is that is a preclinical. That is preclinical means in animals, and clinical means in human. And then it started development, and finally it is approved in 2002. So this is one of a success story of rational drug designing, and I'd like to share this with you. So the first thing, as I told you, is a target identification, target validation. The target safety was not so much for when this was discovered, because this is a kinase, and kinases has a lot of problems. So the target identification started in 1960, to be honest, and then it got validated, revalidated, and many science and nature papers after in 1990, this target was identified as a valid target, and they started designing drug against that. And same, the target safety is something that was not so much existent in 1990s. It is very, very existent today. So there, Imatinib has some problem with target safety. Uh, so, uh, so, so that is all I wanted to talk about target safety. If you have any questions, please ask me to the end of the talk. Then I'll go to the discovery stage, that is the drug discovery unit. The discovery stage consists of a drug discovery unit. These people are responsible for giving us the molecules. Then we have the drug safety unit, like people like me who work in drug safety. And our job is always to criticize the drug discovery unit to say, come up with better molecules which are safe. And then you have the pharmacokinetics persons who are actually, pharmacokinetics is a subject that tells you how our body treats the drug. So when you take a drug, our body, in general, systematically have a response to the drug, and that is what pharmacokinetics is. And this person's actually tell the drug discovery unit to come up with a better profile of the drug so that the body is happy when the drug is taken. And then you have the formulation and delivery. A drug cannot, you can take a drug as a powder, of course, but normally we see the drug in form of a tablet, form of an injectable, and there are many other smart delivery techniques that are coming up today. And then you have the preclinical unit who are actually responsible for doing the animal experiments. in general, organization of the discovery stage platform for drug discovery. In the drug discovery unit, the drug discovery unit, their main aim is to discover molecules. So they have a target, and they will screen very, very small molecules against that, and then they come up with a molecule that we call as hit. And those hit molecules will be then further optimized, and then they are going to get what we call a lead molecule. And then this lead molecule, if everything goes okay, will be optimized, and then this optimized lead molecule can go to a preclinical trial and become a drug finally. So it's a lot of polishing and ultra refinement that goes on throughout the process. So the drug discovery unit, their main aim is to find an efficacious molecule, a very nice potent molecule, and in order to do that, they mostly start with computer-aided drug designing. And of course, before starting with computer-aided drug designing, they have a structural biology that gives us this structure. And then they screen the small molecules. 100,000 is a very low number that I told here. Normally, they start with millions of molecules in a typical screening platform. But if they after filtering those, they will come up with a molecule number of like 100,000 to 200,000, and then this kind of screen it in silico in the computer itself to see which of the molecules that have the potential to bind to the target. And then they will initially start with a few batches of 500 molecules. Now, this 500 molecules has to be synthesized. So this, synthesizing 500 molecules takes an enormous chemistry effort, and these molecules can be multi-step, and they can take an enormous time to get synthesized, but they do it anyway. And then we get hit molecules if some of the molecules bind to this target experimentally, and then from the hit molecules, if the molecules, their profile is good, and if the toxicity is okay, the pharmacokinetics is okay, then we come up with 50 to 100 molecules. This is a practical, that is what happens in the pharma. And then we come up with a lead molecule of around 5, and then we have few backup molecules just to keep in hand. And then this lead molecules undergoes actually a lead optimization. And this lead optimization is, I'm going to show you in the next few slides. And then finally, the project will come up with 1 to 5 molecules. They will have meetings after meetings after meetings, and then they will nominate a candidate for further development. And this is what the funnel looks like. But there is something that is off the funnel, and those are very, very important. And those are the funnel guys, are the pharmacokinetics guys. They always tell the project team, come up with a better molecule, come up with a better molecule, come up with a better molecule. And also guys like us who always stop them in every step and say the molecule has to be safe. That is the first thing that we care about. So it's always one team going against the other team, and what you end up is a super polished kind of gem, a super optimized molecule, and these units have to be independent. And in many projects, we also have the formulation team that comes up very, very early. But this is what happens in a normal funnel of drug discovery. And so it's always criticizing one team by the other team, and there is always to and fro motion that actually goes on. And finally, the aim is to develop a good molecule that is effective, safe, and our body treats it well. So these are the 3 things that we have to take care in the initial stages. Again, I come to this slide. So just a reminder that it takes time. And so I will now, on the next slide, I'll focus on this 2 years actually. That is an amazing speed that they have come up with. I'm going to talk about that. I come to this slide also again. I have shown this before. So I wanted you to focus on this portion because this is where the actual thing is happening. And this is the active site of our target. So if I zoom it, we see it like this. It's a kind of a tunnel. It goes through, the molecule can go through, on the other side. And then if we remove this clay kind of cast, then we can see through the molecule and see how the big molecule is interacting with the small molecule. The big molecule here is is a protein, and the small molecule here is a drug, which is imatinib. And we can see these lines over here. These are actually a representation, of course. They are actually molecular interactions. So there are many kinds of molecular interaction that happens. Molecular interactions are simply the forces that keep the two molecules together. So these are the molecular interactions. These are the forces that can be represented as these vectors or lines. And this is how a small molecule look in the active site of a protein. So I'll come up with step by step like how this molecule is designed and discovered. I will not talk chemistry. I will, but this is chemistry, of course. But let's talk about this is as a building block. Okay, so we don't need to understand chemistry much here, just in terms of building blocks. So this over here is actually showing the molecules in different directions, which is kind of an interesting way to look at a molecule, always to rotate them. So in 1990, when the project team started, they already had this lead molecule. And this is after screening around more than 2,000 molecules, they came up with this little molecule. And they have found this as active against protein kinase C. But anyway, they found it active against the target. And then they wanted to make it more active against the target. So the next step, what they do is they wanted to go here in this space over here that is there in the protein, and then they actually added this portion of this chemical group over here, and this makes an additional interactions with the protein, and more interaction, it means the stronger, the more effective the molecule will be. So if I go here, they come up like this. So they have developed this portion now, and then they found that this molecule has some problem with other kinases, binding with other kinases that can cause toxicity. So they come up with a modification over here now. If you see it here, so they come up with a modification over here, and this is within the time frame of like 2 years, they come up with this one, and then this is the molecule that they have come up with, initially optimized molecule. They gave this to the formulation team and the pharmacokinetics person. In today's world, we will not synthesize the molecule and do the biology and then give it to the pharmacokinetics. And today, we synthesize the molecule and we give it to the pharmacokinetics. Those in tandem with concomitantly together. But it was 1990, so they do it, they do biology, and then they give it. So anyway, so they come up with this methyl group over here in order to improve the off-target effects, and then they give this to the project team, to the pharmacokinetics person to develop, and the formulation person, of course. Yeah.
Sorry, your audio is not clear again, so I had to interrupt.
Okay.
Yeah.
Now?
Yeah, it's clear now. Yeah. There was echo or something. Yeah.
I have to be actually a little bit close to the
laptop. Yeah, probably.
Yeah, that is what is happening.
Okay.
Yeah. Anyway.
Okay. Thank you. It's good now. Yeah.
Yeah. So the project team gave this molecule to the formulation team, and the formulation team says, "Hey, your molecule is not soluble. How can this be a drug for leukemia, which is in blood, and blood is actually 90% water?" So the project team went back and did this modification. So they included a piperazine methyl group that is actually protonated at pH 7, and this renders the whole molecule soluble in water. And this is the final molecule that they came up with. And this molecule become a drug, and that is called imatinib. And if you know, imatinib is a molecule that has changed the cancer field altogether. And I mean, if we see there are interesting plots, before imatinib discovery and after imatinib discovery, the survival of leukemic patients have improved drastically. So that is not the core of the talk today, so I'm not going to show that, but still, I mean, this is how a molecule is discovered step by step. I will go a few steps back. This is where they started with, came up with this to improve the efficacy, came up with this guy, this metal over here to improve the off target, and then they put this one to improve the solubility, and of course also the efficiency. So this is how a molecule is actually discovered step by step. This is a very fast time lapse of 2 years that I have shown in 3 or 4 slides. But this coming to this molecule took possibly a few thousand molecules and maybe 50 to 100 chemists working together. And so these are enormous efforts actually that happens. To summarize the imatinib discovery, started in 1990, they have the structure, then they, in 1 year, they come up with a lead molecule. It took 2 more years to come up with imatinib or Gleevec, and then they did 4 years of biological experiment, and then it went to the developmental stage, and then clinical trial, and finally to the program 22 and 2002. So I come up with the next topic, which is the drug safety. Mm, um, I'm not going to talk about this much because I can talk a lot on this since I am working on drug safety, uh, because this can be a different topic of presentation altogether. Today's drugs are actually safer, I would say, in terms of the scrutiny it undergoes. Typically in a pharma world, we screen around for more than 150 off targets. So there are many off targets, of course, but we screen for 150 off targets at least for one molecule. So one molecule undergoes an assay against 150 proteins. And then we also do cytotoxicity, a detailed evaluation of cytotoxicity. We do a detailed evaluation of cardiotoxicity, which are not only the cardiotoxicity in general, but also cardiotoxicity against ion channels like heart, like sodium channels, like calcium channels. We do nephrotoxicity, which is a kidney toxicity, and then we do liver toxicity, which is in the cells and also in other enzymatic blocks. And then we do neurotoxicity if it is suspected to cross the blood-brain barrier, and there are many more toxicity assays that we do. So this is not an elaborate list. Every project can have its own off-target profiling. Say, for example, for the kinases, we have to do an off-kinase profile. So for a protease program, we have to do an off-protease program. So for COVID, there are a lot of protease programs that is currently undergoing in many pharma. So that is why I mentioned protease. And all this thing happens before it goes to an animal. And so you can understand, so there are a lot of toxicity studies that were already done before it goes to the animals. And the animals, this is the animal that I've shown is not was only for toxicity. It's not for potency. So you can see these are the small rodents, rabbit, dog, cardiotoxicity model, and then to monkeys for many other toxicities. And we have to use different animal models because different animals uh, share different kind of protein homology and similarity with the human. So we go for many kinds of animal. And these things actually mostly happen in the developmental phase, and also some in the discovery phase, of course. And then all these things we do are feedback to the drug discovery unit, who will then change the molecule accordingly, and then come up with better molecules, and then we go and screen them again. And this kind of cycle goes on and on and on. Typically in a drug discovery program, this cycle can go up to 15 times. Before you can get a good molecule. And there are, of course, the other things that I'll talk a little bit later in other slides. Then I come and touch very briefly on the pharmacokinetics and formulation and delivery. So what is pharmacokinetics? As I told you, this is how our body treats a drug. So these are kind of classified into absorption, distribution, metabolism, and elimination. These are the four kind of things that we always seek when we are doing a pharmacokinetics evaluation. I'm not a pharmacokinetics person, but I know just a little bit. So what happens when you take a drug, it is going in your stomach, goes to your gut, where it is absorbed most of the time, and it goes to your blood. This is an oral drug delivery. Of course, if it is injectable, it's completely different. And then this drug has to go and accumulate in a tissue. Say, for example, we are having a drug for some brain disease. It has to accumulate in the brain, and then it has to pass the concerned cells. It has to go inside the concerned cells if the enzyme or the target is inside the cell, and it has to stay there. It cannot be pumped out. So there are a lot of different aspects. Once a drug is potent and safe, then you also have to take care of these things, which is like a nightmare for any project team. So there are people who are actually dedicated to model all this, and they can actually model them. They are pharmacokinetics modeler who actually can predict fate of a drug beforehand and guide the project team. But of course, we need to do a lot of experiments to actually find this. Metabolism and elimination is something that interests me a lot because metabolism is one of the reasons that results in attrition of an already approved drug. There are many examples like that. One of the examples is indomethacin, there are so many. There are flufenac. There are so many drugs that are actually eliminated from the market, I would say, or maybe treated from the market because of metabolism problem. I will talk just a little bit one slide on metabolism problem. I know I'm running out of time. So what is the purpose of the metabolism? The metabolism is most of our drugs are actually greasy, that is like oil soluble, which is oily. They are not soluble in water. So our liver, which is actually the factory of our body, so our body never likes a foreign chemical in the body, right? So they wanted to get rid of it. And the easiest way is to get rid of it is to make a greasy molecule water soluble. And so when it is water soluble, it can go easily out of the kidney through the urine. And so this is the main job of the liver and the liver enzymes. So they identify the foreign molecule and they make a greasy molecule, water soluble molecule, and help it eliminate the body. Um so I will just talk a little bit of the imatinib metabolism that it has a problem. So many drug undergoes this kind of greasy oil soluble and oxidized actually to form a water soluble drug, and then this oxidized product is actually a problem in our liver cells, and it can cause a lot of liver issues and nephrotoxicity and many things. So this is the imatinib that we talked about a little bit earlier. So in our body, there are enzymes in the liver. I intentionally didn't put the name of the enzymes. These are actually CYP enzymes, but anyway, their job is to oxidize the molecule to make it more hydrophilic. But a chemical is a chemical, so it will undergo a rearrangement reaction sometimes. And if it does not undergo a rearrangement reaction, it will be eliminated just like that. Suppose it undergoes an imatinib undergoes a rearrangement reaction and forms a very reactive intermediate like this. And this then interacts or reacts with the proteins that we have in our liver, and those are or maybe antioxidants that are there in our liver. Uh, for the persons who want to know, it's glutathione maybe, and then it forms a linkage with our proteins, and our body does not understand this protein because it is something else. It's not a protein anymore. So our body then initiates an immune attack on that protein to get rid of the protein itself. And so that also causes very hyperimmune reactions, and that can cause serious liver toxicity. And this happens in imatinib and other anti-cancer drugs because they form this reactive intermediate, and this becomes a dangerous intermediate. And that is the reason that chemotherapy or many other, like say for example anti-tumor drug, they have liver toxicity issue. So currently we do these things, this kind of screen before this drug is actually, this molecule will be actually recommended to the project team. We do this kind of studies now beforehand and say, okay, hey, this can have a dangerous metabolite. Beware of this and change the molecule. So we do this now before, but it was 1990 to 2002, so they didn't do this that time beforehand. So So there are many examples like this, a lot of examples. If I start talking the name of the drugs that you and I sometimes take, they also may cause this kind of dangerous um, problems. And these kind of problems are called drug-induced liver injury or DILI. So anyway, with that, I'll just try a little bit of delivery problem. So delivery in general in pharma, innovative delivery is not like there are, of course, innovative delivery unit, but in pharma it's mostly the traditional delivery units like tablets, parenterals, and other things. But since in academia, I was in academia also, so we can use like innovative drug delivery techniques. I will just go on it very fast. So one of those delivery systems is called responsive delivery system. So you know that the cancer cell, the pH of the cancer cell is a little bit acidic, so we can develop a drug and a polymer so that it will release the drug in the cancer environment and not release in the normal environment, and thereby causing the less toxicity. The delivery is actually the drug. If you take it, swallow it through mouth, and this is where the thing is happening. So the drug has to be delivered here. If it distributed throughout the body and diluted before it goes to the receptor, then there is a problem. The efficiency goes down significantly. So in order to solve the problems that cannot be solved while discovering of a molecule, delivery people actually try to solve it by targeting the drug to the particular place or a particular physiological response. So I have been working on this responsive drug delivery system for quite a bit. I'd like to share just one of my work that is still under review. Hopefully, it get published very soon. So this is the latest paper I will publish, and possibly I will not publish for a long time after that. So this is a polymer. There's beads over here, and this is a small molecule drug, which is gentamicin, and it's an antibiotic. So you can also actually stimulate drug polymer interaction. Since the drug delivery paradigm has entirely changed, it was not a traditional way to call the designers, the molecular modelers in the drug delivery unit, but our expertise have now little bit increased, and we can now do some simulation for in the drug delivery space also, and that is an exciting thing that is going to happen. So drug delivery is changing, and this is very this is an excellent field for people with any background, maybe a biologist, maybe an engineer, computational guys like us, and chemists, of course, and definitely the pharmacists. So they all have enormous opportunity in drug delivery, especially in academia. So with this, I will kind of in my talk. And so what I want to say is in a drug discovery and a delivery platform, and it is an exciting field. It's a very challenging field, but an exciting field. I hope my lecture inculcated some enthusiasm about the student to take up drug discovery or delivery as their future endeavors. And I'd like to thank a lot of people. So my mentors like Professor Arijit Basu and Amira Golu. Uh, so they are like wonderful. I was very fortunate to have joined Daniel Anderson and Robert Langer at MIT, and I have a lot of competition around. So I thank a lot of high computing facilities. All my friends and colleagues at the University of MIT and also Takeda currently. So yeah. And this is a little award that I got at Program for Advanced Technologies, which I'm also the editor for now. So
Dr. Basu, the audio isn't clear again. Sorry.
Yeah. So this is So now it's okay.
Yes, now it's okay. Now it's okay.
Yeah. I'm kind of forgetting to come close to the laptop and talk. Sorry. So this is a tiny award that I got last year. with, of course, Professor Mazachevsky, who is a very well-known person in the field. And we also have a company in India, actually, so which we call Biomolix Innovation Private Limited, where we make hydrogels for mainly burn wounds and other kinds of wounds. And this is like a protein-based hydrogel. And so this is also exciting. I'm thankful to the Biotechnology Innovation um, to fund this, though we are struggling now due to the COVID situation. But I'm still hopeful that something is going to get out of this. With this, I thank all of you for attending this, and please ask a lot of questions. Thank you so much.
Thank you, Dr. Basu. I think people will listen to you because we do have people from both platforms, which is the Google Meet and YouTube. A lot of people streamed in, and we do have quite a few questions. So we'll try to get to as many as possible. And if you're willing to answer them, we might not get to all of them. So we'll try to take as many as possible.
Sure.
Yeah. So the first question, sorry. Sir, in spite of such a, it's by Otonu Banerjee. Sir, in spite of such a meticulous step in lead molecule optimization, side effects of some drugs could not be avoided. Please explain this part, why side effects are obvious for many such drug molecules.
Well, Otonu, thank you for this important question. There are, I'm audible, right?
Yes, you're audible now.
Yeah, thank you for this.
If you could also, I don't know, people would want to see you. So if you could stop sharing your screen and turn on your video.
Okay. You guys can see me, right?
Yes,
yeah.
yes.
I can see me. That means you guys can also see me. So thank you, Otonu, for this important question. We are trying, and a drug can actually bind to off target. This is one of the reasons for showing toxicity. And there is another thing that is extremely difficult to determine. That is what we call idiosyncrasy. Idiosyncrasy is a drug molecule behaves differently when it is given to me and will behave differently when it is given to you because of varied genetics, because of the protein expression that you have and protein expression that I have. A drug can completely behave differently between persons and also between populations. So that is something that even if you do some meticulous evaluation, and today's drugs are definitely way more safer than earlier drugs, still this is something uncertain. And there are many drugs which, if you see, that are withdrawn because of this problem. And one of the major reasons of the drug being withdrawn from the market is idiosyncratic behavior of a drug. Say, for example, diclofenac. Diclofenac is an is an NSAID, which is you take when you have pain anywhere. Volini, I think, is one of the brand names. So diclofenac can cause liver damage to some people, if you take it a lot, but it is perfectly safe for others. So there are many other molecules like this. When we were little, we used to take indomethacin, and now it is withdrawn. We used to take nimesulide, if you remember. It's withdrawn now. So these are like idiosyncratic problems that is extremely non-linear to predict. Even if we include a lot of different population in the clinical trials and a lot of population in the cell-based assays, this idiosyncrasy is something that is actually can kill a drug molecule. So this is the major reason for attrition of a drug. There are some side effects, some necessary evils. Say, for example, we are developing a drug against cancer.
So there are some side effects, necessary evils that we cannot do much about because getting a drug is more important than those side effects. But say, for example, we are developing a drug against a cardiac issue. Say, for example, we have developed statins. And statins are relatively safe, but not safe, of course, because they can cause liver problems. They can cause type 2 diabetes within 1 or 2 years in some patients. So these are idiosyncratic because it does not cause to some and cause to some. So it is also the patient population variation that we have to take care. I think I have answered your question.
I think so. Yeah, that was very well explained. Yeah, thank you. So the next question is from Papri Bosak. What about checking about cytotoxicity in human-specific protein interactions? Is checking in cell lines enough in that case?
Of course not. Of course not. And we don't do it only on cell lines. We go for many preclinical evaluations. Preclinical evaluations are actually going from small animals to bigger animals. But of course, if you go to a cyano monkey, they have a lot of proteins that are different than ours. Cytotoxicity, we don't do a single type of cell lines. We do a lot of cell lines, and also we go for specific protein assays that are actually developed, and over the period of time, actually, we have the good thing about drug discovery is we have failed a lot. Because we develop so many molecules and we always have a reason for that molecule to fail. And once we know why we failed, we can always go ahead in the next discovery program to correct it. So we have a list of proteins that are actually culprits for withdrawal of a drug molecule in the program. And so we do first a protein-based evaluation, we do cell line-based evaluation, and then we do preclinical evaluation. And most of the drugs, if it is not oncology drug, that is, if it is not cancer drug, we do a pilot phase 1 safety trial also on humans. So there are a lot of screens that actually goes through for a drug discovery program. But sometimes we miss something. So that is what it is. I mean, we cannot help it. Sorry.
Thank you. The next question is from Anjali Kulkarni. Which are the latest protein targets that are being studied currently to screen for anti-cancer drugs?
See, I cannot talk about the protein targets that are happening in my company. I cannot say that. Okay, so let's talk about different types of modalities. So that is quite exciting. If you want to be an anti-cancer in the anti-cancer field, I will definitely tell you to go for immunotherapy. Immunotherapy is actually happening, and that is possibly the best answer to cancer, I can say. So for immunotherapy, you go for antibodies mostly, and then you have the cell therapies, which are very exciting that are coming up. So these are the two fields I will say in cancer that are actually hot. One is immunotherapy using small molecules and antibodies, and then you have the cancer field. There is another field that is actually quite important, that is called PROTACs. And I don't know if you are aware of PROTACs. PROTACs are actually protein degraders. So these are not small molecules. They are two molecules actually. One molecule fits to the drug, and the other molecules actually recruits ubiquity to actually degrade the target altogether. So that these molecules are very, very extremely potent molecules, picomolar potency. And this is also an exciting modality in oncology that is coming up. So, and there are, of course, a lot of targets. Every day we have a new target. And kinases are something that is, of course, VEGFR and PTGF and Abel kinase, I told you, they are still hot, actually, the kinase targets. And there are also other targets that are also hot. But mostly it is the immunotherapy targets are actually super hot, I would say now.
Thank you. The next question we have from Shoptok Banerjee. A drug is made to bind to some receptor, but as we know during cancer, the receptors undergo spontaneous mutation and drug fails to bind. So later stage, the drug is not effective anymore. Is it possible to cope with this problem in future drug discovery?
So this is the problem with drug. Cancer cells are way more smarter than our normal cells, and they don't like to get inhibited by a molecule. And currently the clinical strategy is to attack this cancer cells from various directions. And of course, cancer is a, is a challenge that is yet to be solved. And so so currently, what is done is, say for example, Imatinib, it has a lot of resistance because of the mutations in the cancer cells. So they come up with a better chemotherapy, and normally the clinicians actually change the chemotherapy to an advanced one. They start with something that is very very responsive, and then they come up with next stage of chemotherapy, and then they go on changing it. So that is the current. That may not be an ideal situation, but in drug discovery, of course, this is what we always do. So we always see the point mutations and we develop a newer molecules against that. The only problem is it will take time because it has to undergo clinical and everything. So I mean, in delivery front, sometimes this can be tackled using nanotechnology and other things. Of course, there are many other chip-based delivery techniques that is developed by in my previous lab, that is in the manual lab, that they will take the cancer cell out and they will actually what do you call it, harbor them in a chip, and then they will put different chemotherapy to see which chemotherapy the cell is actually effective against, and then they will choose the chemotherapy design according to that. So these are investigational, nothing practical yet. But yes, of course, we do drug discovery by taking into consideration the mutations that are happening over time. But it takes time, of course.
Of course, yeah. If you're okay, should we take a few more questions?
Okay, go ahead, no problem.
Okay. Next is from Kakoli Bhadro. There are two approaches in natural product-based drug discovery. Forward pharmacological approaches and reverse pharmacological approaches. Which one is more preferable?
Both are actually used, to be honest. One is, we call it a phenotypic screen sometimes. That is, we put the molecule and see a phenotype, and then we try to figure out what is actually causing the effect. This is one way. The other way is to start from rational designing and go ahead. So typical this phenotypic screen and this kind of reverse pharmacology thing is actually was done before in natural products, of course, and it's still ongoing. I have worked in both, to be honest, but I find as a designer, um, reverse pharmacology is very difficult for me to understand because you should see the number of experiments that you need to do in reverse or forward. The number is almost similar, so possibly more in reverse. It has a specific advantage or not? I mean, I will say whatever works. If reverse works, reverse is great. If reverse does not work, then reverse is not great. So it depends on the project actually. So the presentation that I showed you is a forward pharmacology technique, which is based on rational designing. This can also be done and has been done for natural products. And so I mean, whatever works for development of the molecule, I will say. It depends actually. So it's a very highly subjective project-based question. It's not a general question to answer, right? But it's a very important question.
You've partly answered this, so I'll let you decide if you want to add to it. Is from Zoo Fish and Kazi. Drugs are also tested for specific allergic responses and other drug interactions. Can you briefly describe how it is done and during which step of drug development?
Well, it is mainly done by the pharmacokinetics persons. So it is done after the candidate has been nominated. That is after in the interface between lead molecule optimization and to the next stage where we do mainly GLP studies, which is like good laboratory practice studies for the development and going to higher animals. So it is done. It starts from there and it's done throughout. And it is also continually being done after the drug is marketed. So it's called therapeutic drug monitoring. So after the drug is in the market, it is still done. So we can have a drug that is like 30 years old and still it's been monitored. So we still get data from 30-year-old drug that is there in the market. It's done in every stage.
We'll probably take two more questions if that's okay before ending.
Yeah, sure.
So the next one is from Piyali Chatterjee. There are drug patents that scientists in academia make. How is the process different with such patented drugs, or is it not?
Now, patent is a very geographical and a very, what do you say? It's a country-to-country variable legal right. So if we do a patent in India, so you get a completely different right than you do a patent in the United States. Typically, when I work in a lab before this, where every step we patent, we mix one solution into other, got a red thing, we patent it. We took that red solution, we put it in a blue solution, we got some other color, we patented it. So I work in a lab where every step is patented. So now MIT has spent a lot of money on patents. So is it useful? Of course, it is useful. This actually prevents others from breaching your intellectual property. So yes, and in academia. So this is an academia. I'm telling you. So the aim of my previous lab, the Langer Anderson lab, is to get this product to the market and to sell it to big pharma or small pharma, and to get what we call a small biotech out of it or a small pharma out of it. So there is a commercial aspect that is always involved. So I would say when the work that I have done at MIT, that resulted in possibly 4 or 5 patents for a single project. It covers from all the aspects. Sometimes it is too much, but possibly it is needed to actually protect your intellectual property. Some scientists don't patent, so they wanted to give their science away almost free. So that is also another way that some scientists do follow. Should you patent or not? If you think your molecule has, or your research has a commercial viability, then of course go ahead and patent. So that is my suggestion in the patenting space, because I have seen scientists who have lost money for not patenting, and I have also seen scientists who have patented a lot but never got a single money out of it. So I have seen both. It depends on the research, of course.
Thank you. So the last question, actually, we'll ask, since we were talking about clinical trials and it's COVID.
I don't know much about clinical trials, but anyway.
No, it's not about that. I'll let you deal with the question. So this is from Shoptok Banerjee. He says, as you told, it takes years for a drug to pass the safety measures before it comes to market. How are companies bringing COVID drugs in months? Is safety compromised?
Safety will never be compromised. That is our policy. We cannot compromise on safety. If it takes time, it takes time. The thing that is done by the companies, I mean, I'm not sure but anyway, the thing that is done by the company, say I work for a company that develops a COVID drug, okay? So what happens is the companies actually put enormous manpower into this effort, and we don't evaluate this molecule in the company itself. So what we do is we outsource this kind of research, to companies, to a class of companies which are called contract research organizations, also called CROs. And so the CROs actually have also stepped up. And so there is enormous money being poured into this research. And in some way, a research that will take 5 years may be shortened to 3 years. Because, say, for example, a typical project will have like 15 chemists working on it. For a COVID project, it may be 30 chemists working on it. So they develop the molecules faster. They do the evaluation faster. And so maybe 5 years can be reduced to 3 years, but it is not compromised. It cannot be compromised because that is a policy of a company. So if you have to compromise the safety, you have to change the policy of a company. That is not possible in a big pharma. It is impossible. So I will never say a safety will be ever compromised for a drug in the market because those are independent teams, and pharma have mostly done whatever they can do to ensure safety. But sometimes, scientists also are human; they do mistakes.
For sure, for sure.
Yeah.
And I think it's more important now that people are realizing the signs that scientists make failures, and that's how we learn.
Yeah.
And so, yeah.
We fail all the time. I mean, we fail, I don't know, 999 times, and maybe succeed once. I mean, I don't remember a real success in the drug discovery in my lifetime yet. So, so most of the drug discovery scientists, they fail. And that is where the excitement is also.
Yeah.
We are not afraid of failure. So, yeah.
That's good. Yeah, thank you. Thank you for answering the questions. We couldn't get to all the questions, so apologies to participants whose questions we couldn't get to. But thank you for answering all the questions and explaining like what time and effort goes into drug discovery. And thank you for actually making our drugs safe. Thank you so much.
Thank you. It was a real pleasure.
Yeah. With that, I'll actually ask Professor N.R.A. Banerjee to end the session. But actually, before I do, I'd like to announce the speaker for next week is Dr. Saurav Ghosh from Yale University School of Medicine. So please join us next week. And with that, I'll hand it over to Professor N.R.A. Banerjee to end today's session. Thank you, everybody.
Thank you.
Thank you, Sanjana. Thank you, Arijit. I enjoyed your talk so much. It's
thanks.
almost your talk is like a ready reckoner for anybody who wants to know about drug discovery and drug development. But as you rightly said, it's a long process. And best wishes to your company. And congratulations for winning the grant from BIRAC. It's a highly competitive grant, so I hope you're doing good. And yes, lots of work goes into it. And for Shopstock, I think, who asked about COVID, there's something called repurposing of drugs. So what Dr. Basu now talked about, that millions of dollars are pumped and manpower hours are pumped into the discovery and the development process. And the pharma industry would always like to get as much mileage out of it as possible because the safety issues in the formulation have already been taken care of in the approved drugs. So repurposing is also an important area. And because Arijit is a computational chemist, I think a lot of the in silico probability of failure or success, a lot of the in silico predictions also happen in your hands. So a big thank you. And Mohuadi, if you're listening in, thank you to you too. I'm so glad I had tuned into your talk, and we must have more of these talks. Which brings me to that, you see, I throughout the talk, and it's so engaging but I see in the feedback links in the chat box a lot of people asking for feedback links, which means you're here for the certificate. Now, what I would like to stress upon is that what I said at the beginning of the talk is that you see scientists with years of work, under their belt, they are spending an hour or more discussing their work, answering your questions, and that is the biggest, takeaway from the talks. And when Arijit took so much time to answer your questions in such great detail, I think, just the feedback link or the asking for the certificate sort of undermines the whole purpose of this webinar. So that's not very nice. But for the enthusiasts out there who asked so many, Kakoli I saw ask questions, so for those people, this is a message that I would like to give, is that it would be great to have speakers like Arijit with real training and insight into the process. And PPP is also trying to facilitate collaboration, as you rightly said, Arijit. The trainings and the collaborations. What we cannot do, we rope in other people who can do it for us. And especially when you talk about contract research and or translation, we look for people who can take care of the downstream processes, such as taking it to the market, the little things that matter, the regulatory brochures, the DPRs and the transcriptions and safety issues, and there is so much available. So PPB is also trying to facilitate that. And for the people who actually engaged in the talk, this is something that I would like to say is that we would like to organize a panel discussion where real networking and collaboration can happen. Because you see, Arijit, right now when you were talking, you're like a class. You're talking. It's a one-sided thing, and we are listening to you. And so after a round of talks by experts from different fields, if we can have a panel discussion, and then we can actually talk about hand-holding in terms of translation, in terms of real products or processes that can come out through the kind of collaborations and networking that you just spoke about. So there are the pharmacology people who are working on the development aspect of it, the safety aspects of it, the CYP inhibitions and the liver microsomes and the PKPDs. And so that is an important aspect. Then you have the synthetic chemists working to fine-tune the molecules. And then you have the biologists. And, overseeing all three are people like Dr. Basu here who will say, no, this is not going to do. And I'm going to put this little, what did you say? Was it your daughter who said that what you see on the screen? I heard that those funny designs that you're playing with all the time. These are molecules, and you're studying the dynamics of it. So he sort of oversees all these things. So we should probably have a panel discussion, and we'll probably also send a feedback link to the speakers for suggestions that how better we can use the platform, since a few hundred people were listening into you right now, and other people will also be tuning in. So let's have some real stuff coming out of the discussion. So thank you so much, Arijit. It was
it's a real pleasure.
It was excellent. Thank you so much.
And I'm really sorry that I cannot take all the questions.
For tuning in.
Yeah, I'm really sorry I could not take all the questions.
Send you some of these questions.
Yeah, please send me offline the questions. I will try to answer this. Like I will try to type them up and send the answers to the questions.
Kind of you.
Yeah, no problems. I mean, I was a teacher once.
If you have any further questions for the speaker, write an email to PPB, and we will pass it on to the speaker. That's very nice of you. Very, very kind.
No problem. Absolutely fine. I may be a little bit late, but of course. Yeah.
Great.
Okay. Thank you so much for having me.
And tonight,
yep.
Pleasure was all ours.
Mutual. Enjoy your dinner, all of you. Thank you so much.
Morning at your end.
Yeah.
It's morning at your end, Arijit. So I'll wish you have a good one today. And for the rest of us, because people are tuning in from all over the globe, for us, it is good night. So people in India have a good night.
Yeah. Thank you. Thank you all. Bye.